Okay, here we go. So the title is about neuroinformatics and replication. So I put two subtitles under the title. So we want to beyond bash scripts and Werner's curse. How? I think I will bring you guys back to the theory of reliability and validity, right? So I heard someone talked about, well, yeah, in Red's presentation, we really need to thinking, and not always take big data and calculate, right? So the outline I'll give you. I think the first two items I don't need to talk about too much. You guys already gave that, and I will turn into the neuroinformatics related things about software tools and reproducibility. And next I will talk about the statistical theory of reliability and validity. And especially I will focus on test-retest reliability, and I'll give you guys a very comprehensive review about it, particularly in neuroimaging field. Next, I will introduce you guys a big data of test-retest reliability. It's called CORE. It's Consortium for Reliability and Reproducibility. And finally, I will talk about a little bit about the solutions for reliable human brain mapping in future. Okay, I started to learn population neuroscience when I read this paper. So which one is the pointer? Which one? The red one? Okay. So I read this paper, it basically says population neuroscience is trying to sort out how the genetic and environmental factors affect the human brain development, right? And also, last year I got the book called Population Neuroscience from Tom after the conference in Nanning. So I get to know this field. So it is interesting to see that this field is ready for neuroimaging. Neuroimaging, we get a lot of data sets by now. We get a lot of data now. The data just keep coming, right? And you can see, you can visit this list of the big neuroimaging data from here. But my personal feeling about this data for this field is we really lack or missing some meaningful, unique analytic tools for big data. Right, we still use those softwares, not specifically designed for big data, right? But if we review these literatures, we can see in economics and social science, they already have some tools for analyzing big data, right? But we are not there, I think, in neuroimaging field. So I think we really need to think about the reason why we haven't there. So next is about software and reproducibility. We have so many software toolboxes, right? You can use any of them to analyze your data, analyze those big data, right? The very, very naive question is about when you change your software tools, how about your result and finding will change? Right? We don't know. Right? This is a kind of a reproducibility of the findings across different tools. Right? I list some issues here related to the software and reproducibility. We just don't know. So we already talked about a little bit validity, right? And I will give you a different perspective from clinical side, right? This is a kind of a very straightforward description of individual differences, right? So, for example, if we focus on a specific disorder, and one type of individual difference is directly related or caused by the disorder, right? Another is related to the individual, but not specifically related to the disorder, right? But the third part is the random errors, not specifically related to the specific individual. So validity is the portion of disorder related variability, right? And reliability is the individual related, variability proportion. So you can clearly see validity always less than reliability or smaller than reliability, right? So, but unfortunately, we never can, measure or get this validity, right? So normally we estimated reliability as a upper limitation of the validity. So from clinical side, we first ask the question about what about the reliability of the specific tool or diagnosis tool, right? That's the problem. Yeah, you already presented this, right? So we all want this, right? This is a reliable and validated methods or tools. This is the worst thing. We don't want it. It's unreliable, unvalidated. This one is reliable but not validated, right? This one is validated. Averagely speaking, it should at the center, right? But it's not reliable. This is a different terminology related to accuracy and precision. Yeah. So all the factors related to reliability is individual differences. There are two different types of individual differences. So I'll give you an impression about this. So this is the same individual measured across 10 different occurrences. And this different color means different individuals. So we have intra-individual variability or intra-individual differences as measured by the variability for specific individual across different measure occurrence, right? This is called intra-individual variability, and we have inter-individual variability across different individuals, right? So this is two different types of individual differences. So reliability is calling this. The high reliability cause very small intra-individual variability and very big inter-individual variability, right, from this equation. So I love equations, but but my ground is mathematics. So I'm very happy to see Anqi's slides, has a lot of equations, but normally I don't put too many equations in my presentation. But I put this one here, right? And also in this field, the tools of computing ICC, the reliability now are available. This is a toolbox from Afni's group. They just published this paper this year, I think. And now in Afni, you can calculate the ICC by using repeated neuroimaging measures. So why ICC is so important? In theory, we know this is the true signal. We never can get this true signal clearly. It's always polluted by some noise, right? And we measure noised signal. This is for example, this is for two different variables, right? After we calculate the correlation between them, we always calculate the polluted correlation, right? We never know this true correlation in real life. But we can see in theory there's an equation, there's a relationship between these two. And we can see the correlation between two variables never beyond their reliabilities, right? For example, if you have a unreliable measure, you never have a chance to get high correlation, right? That's the theory. So what is the real life? So look at this paper, right? But we saw a lot of high correlation in publications, right? Many of them published in Science, Nature, and whatever, very high impact journals, right? Look at this. They got the correlation between task, what kind of task activation, and the what is the social distress measure or score. This correlation is far beyond the what we expected, right? That's wrong, right? We all know the task-based fMRI. We know that their ICC about 0.7, right? The behavior measures is 0.6 to 0.8, something like that, right? In theory, how you can get this, right? Yeah, if you get more plots there, and you will reduce that correlation, right? So there's another theoretical consideration for reliability. So about the correlation, this is the recent paper published in Behavior Research Methods. Actually, there's a fact that most robust cognitive tasks do not produce reliable individual differences. Why? Because all the cognitive tasks designed by people, right? So when people design those tasks, they want to introduce individual differences for each group, right? That means you heard the individual differences. Also, that hurts your reliability, right? We know our reliability wants high inter-subject or inter-individual differences, right? So that's why this is very scary, let me think about all the papers published previously, right? Look at this. This is the true correlation we want to detect the correlation. This is the reliability for the two different variables. We can see when the variables are very reliable, this is the actually observed correlation, and in this case, you really see we don't need big data. We just don't need the big data. We need reliable methods, right? So, but if the reliability, reduced like this, right? Here, for example, here, even you want to observe 0.3 correlation, you can see you really need so-called big data, right? So the reason behind of people going crazy to get big data is originated from reliability, the poor reliability people. Just stop before that, right? So that's the reason. So next few slides, I will introduce the reliability, especially for test-retest scans across different neuroimaging modalities. You can clearly see how we get regarding the reliability for this field. Right. For test-retest reliability for structural MRI, we got the measures like thickness, gyrification, or this fractal dimensionality is some kind of measure, right, to measure the complexity of the brain geometry. They used two different test-retest data sets. So one is from they call big data. Another from what is that? That's Xi-Nian structure, something like that. I can't green or something. This is big data from Randy Buckner's group. You can see for these structural MRI matrix, the reliability is good, right? It's pretty high, right? And this is also for different areas of the cerebral cortex. And this is for the subcortical structures. You can see, right? Oh, that's for that. Those are for structural MRI. How about the task-based functional MRI? You can see this. This is the ICC value distribution. The average ICC value is 0.4, right? That's really bad, I think, right? And even the peak of this histogram is still less than 0.6. So how you can get the correlation of 0.8, right? So that's the other problem. So for resting state fMRI, I spent, I think, 12 years to work on this, to see how these measures regarding their reliabilities. You can see this is across different measures, blah, blah, blah. And I published these papers and for different resting state fMRI matrices, right? And then I don't want to show the maps from each paper. I just did a meta-analysis based on those maps of ICC, and to see how reliable these matrices. So the big area of this spider graph means higher reliability. This one is good. This one, regional homogeneity is good. And this hemispheric connectivity is also good. The SA is good. But what happened to this, right? This commonly people keep using seed-based functional connectivity, right? Their reliability is really not good, right? This is why I want to show this here. So those are meta-analyses based on the small sample of test-retest data. And I reanalyzed the ICC values based on the HCP test-retest data. And we kindly get, these are ICC maps, basically replicate the finding, right? Some of those resting state FMR matrices are really, really reliable. For example, this, right? Very stable. And some are really bad. For example, this one, right? So I think these provide some guidelines for the field to use these matrices or these techniques, especially for population neuroscience, right? We want to detect those real changes. So, I think this is the reason why we share the big data of test-retest data for neuroimaging. This is called Consortium for DABAR, the reliability and the reproducibility. And now I think this field really get enough data for test or validate the reliability of different methods. Right. Okay, this is the paper we published, and I'll give you a list of further reading about reliability. That's the one. Before we plan to launch this consortium, this is the consortium data paper. We have another hardcore consortium paper is undergoing. It's very hard to get published because this is really, really challenging this field. So, and also this is some news is interviewed by Nature, and we described the motivation why we do this. So once we have the big data, so how this will bring to this field, I mean stimulation, would really give something different. So this is the first one we published. This is the one typical data set we have in call. You can see there are 30 participants, and each of them was scanned 10 times within 1 month. That means every 3 days, the participant will climb into the scanner and get scanned. That's kind of, but this one is for a single scanner. This is a specific scanner. So this will give you a chance or data to extract the intra-individual differences of variability and the inter-individual variability, right? So this is the maps of how the different types. This is the intra-individual variability. This is the inter-individual variability. How these maps looks like. This is very interesting that you can see this is different networks. Oops. Right? We can see for intra-individual variability, really these primary or low-level networks or areas show high intra-individual variability. But for inter-individual variability, these high values of variability appear to high-level cognition-related areas. That's very interesting. So based on this data, Thomas Yale's group developed a new individual level postulation of the brain. People talking about individual level brain postulation, but never using repeated measures, right? How that can be called as individual level postulation. So if you integrated individual level repeated measures, you really get different results and also improve the performance of postulation. You can see this is the 17 network solution, and this can be extended to postulate the individual cerebral cortex. And also this one actually replicated the findings we had in the original paper, right? So also some people using the data I just described to answer the question how long of resting scan could be enough for get reliable or meaningful measure. This group from NIH, I think, get answers such as 7 minutes. Right. This is a solution for one specific metric, not for all metrics of resting state. For example, for functional connectivity, no, I think some people published papers in Neuroscience said you need 2 hours, right? But I think there's a reason about reliability. So also some group using these repeated measures to develop voxel or vertex-based structural connectivity method. This is really cool. We don't know until now. This is the only one can do the white matter connectivity for vertex, for voxel or for millimeters resolution. Also, they showed their method algorithm are very reliable. This one again for structural MRI. I think there's the same data. There's the 30 participants. For each participants, these plots give you the raw data of the estimation of thickness or other matrix. There's 10 plots for each subject. This is really show you the raw data of the repeated measures of brain morphometry. So I would like to talk about several solutions, not the final solution, but the field need to do this in future. The first one is we want as many as people to get involved in this reliability issue to this field. And for this purpose, 2 years ago, we launched a research topic about reliability and reproducibility in functional connectomics. We are going to close this research topic this summer, and there will be 12 papers about reliability to show how we can fix or improve the reliability are just presented in this talk, where those metrics are not that reliable, but you can improve that by proposing new methods or computational solution for them, especially for brain graph theory. So, yeah, this is I think this is one of the final solution. So we really want to extract the standard estimation of those inter and intra individual differences across different scanners, different methods, different time, all those confounding factors. So we launched 3 projects. The first one is called Local Travel Project. We have 3 scanners around the institute. 2 3 Tesla and 1 7 Tesla. So we have 200 healthy subjects. We will travel across these 3 scanners. They will be measured 5 times. This will give you, for the sim participant, how their measures of their brains will change across different scanners. Right? But the scanner sample size is small. It's only 3, right? So to generalize this to more scanners or to the scanners in China, so we have 5 graduate students. They will travel across 40 scanners in the country. Right? This one will give you the individual differences related to the scanners, or how we extract that after we have this data, and we do that. And then also we want to see the really intensive variability of individuals for a scanner. So we have 8 institutional staff, right? Yeah. So they will be scanned 4 or 5 times every week, and across the 2 years, and you will get very intensive data, repeated measures of their brains. Right? I think once we have these kind of data, you have the almost the final solution of extracting or tracing the individual differences, and at that moment, we really can get the maps of the individual differences. So finally, actually, we are planning a big project for twins, for lifespan twins. Right? This is the design. This is a accelerated longitudinal design. So we will scan 6,000 twins in China across right now is about 12 provinces. Right. So I think that's all. I still have. No, I have 6 minutes to answer questions. Okay, so I would like to thank all the lab members from my group. So this is the picture I just took last night. Thank you. So any questions?